introduction. And I'll have the great pleasure to introduce our speaker today. We have a full house this morning. So welcome to the first external speaker of the fall seminar series for the One Health Microbiome Center. And there is no better way to start off this way than with our colleague from the University of Chicago, Dr. Ron Blekhman, who interconnects human health, hologenomics, and the causes that shape human genetic and microbiome variation. Ren is exactly the kind of mid-career scholar who others might want to exemplify. Deeply productive, collaborative, and impactful, whose scholarship is woven into the fabric of the human microbiome sciences. We followed Ren's career for almost 2 decades. He earned his bachelor's in computer science and biology from Tel Aviv University in 2005, PhD in human genetics from University of Chicago in 2010. He then joined Andrew Clark's lab to do a postdoc at Cornell and joined the faculty at University of Minnesota in 2013. He's been an admirably humble and influential force of nature in the human microbiome sciences since then. And in 2022, he joined the faculty at University of Chicago as an associate professor, where he resides in the section of genetic medicine within the Division of Biological Sciences. Ultimately, and as you will see today, Ren's work is a powerful voice of creativity and innovation in the world of microbiome data, spanning human genetics to microbes. Not many of you know, but he's also a very talented tweeter. Last week, this talent was on full display, and I think all of you can relate to what he's doing here in the twist of a tweet. Hello, I'd like to invite you to do my laundry for me. If you accept, I ask you to do my laundry in the next 7 business days. If you decline, please give a reason and recommend 2 to 3 colleagues to do your laundry for me. So with that, Ren, we have a little token of appreciation plaque. I have the box here.
Thank you so much.
Thank you, and this floor is yours.
Thank you so much. Thank you everyone for coming. Thank you for the very, very kind introduction. It was like the first time someone read aloud one of my tweets. Not meant to be in this medium, but it still works. So thank you everyone again for being here. I'm happy to be here. I think this is one of the places where I'm giving a talk and I don't actually have to introduce the human microbiome. I'm sure you all know what it is. Just in a few words, maybe like 30 seconds, we all know what the microbiome is. There are trillions of organisms, you know, several times more than human cells, thousands of different bacterial species, millions of bacterial genes, different bacterial genes in our microbiome, which for me, I think this is the most interesting tidbit because it's kind of coming from a human genetics background. I know we have in our own genome, we have like 20,000 genes. Knowing that the microbes have a few orders of magnitude more is something it's for me just really exciting to just think about the functional potential of these microbes. We know they're found all over the body. Mainly what I'm going to talk about today is the GI tract. And then I think probably my favorite tidbit is like the weight of these microbes combined is about 2.5 pounds, which is something I keep in mind whenever I step on a scale. And why is the microbiome important? Again, I don't really have to emphasize this because you all know, but microbes are involved in metabolism. They can metabolize all kinds of compounds our own genome cannot. They interact with the immune system. They can train our immune system, and they also provide protection against infection. And like you all know, they've been associated with a lot of different diseases and disorders. This is just a figure from a review paper by Kinross et al. Just showing some of the associations that were found between the microbiome and different health conditions. Things like colon cancer that I'll mention later, obesity, metabolic syndrome, inflammatory bowel disease we're working on. So lots of different autoimmune disorders, lots of obesity-related disorders, things like that have been associated with the microbiome. The paper that's now like, I think 10 years old. So since then, basically any disease you can think about there's a paper showing it associated with the microbiome. And again, for me, coming from a human genetics perspective, this is really interesting because in the last few decades, there's been this huge explosion in studies trying to find the human genetic basis of diseases. And this was usually done with genome association studies. This just shows kind of the human genome. Each of these lines is the chromosome, and each of these little circles basically represents a region in the human genome where there was found to be associated with some kind of a complex disease or disorder. And the important thing to take from here is if you look at this list of these complex diseases that have a human genetic basis, there are basically the same diseases that are also associated with the microbiome. So things like digestive system disease, cardiovascular disease, metabolic disease, immune system disorder. These are all diseases that have been associated with the microbiome, as well as they have a human genetic basis. So basically what I'm saying is you have the same diseases have host genetic basis. So human genetic variation in our genome is associated with those phenotypes. And then we also have an association with the microbiome. We don't always know what the direction is, but there is an association with some kind of a change in the complexity of the microbiome with these diseases. The challenge, I think, is that almost all studies that look at the host genetics of diseases don't actually look at the microbiome as a component. And vice versa, most studies of the microbiome in human disease don't really think about host genetics as something to incorporate in the study. In my perspective, if we actually want to understand the basis of human disease, the basis of human phenotypes, you will need to consider both of them, both of these, both host genetics and the microbiome. And I think that first step is to actually characterize the interaction between host genetics and microbiome, to understand how they work together. Are microbes controlled by our own genes? Can we identify the certain microbes that are controlled by host genetics? Can we find the host genes that are associated with changes in the microbiome? And then maybe can the microbes actually control the expression, the regulation of some of our. So basically, this is kind of the basis of all of the work that's done in my lab. We're basically really interested to look at those interactions between host genomics and the microbiome. The first step, I'll tell you about some studies we've done looking at the association between host genetic variation and the microbiome. I'll start with just kind of a little bit of a background about some of the earlier studies from my lab looking at common genetic variation and the microbiome, some disease-causing genetic variation. I'll just kind of mention some studies that we've done, so kind of lay the kind of the foundation from some of the work I'm going to talk about afterwards. Then I'll tell you about two stories and spend a little bit more time talking about these. One, talking about the heritability of the microbiome, a study that we did in wild primates. And then a new study that's still ongoing. We just had a preprint very recently out, talking about global variation in the microbiome, differences in the microbiome between human populations at kind of a global scale. And then if I'll have time, I'll talk about this other direction that we're just kind of getting interested in, which is once we know there's a lot of variation in the microbiome, we want to understand how it affects the host, how it affects disease. One way we think this is happening is through changes in host gene regulation. The microbes can affect the regulation of host genes that are interacting with the microbes. And I think I'll try to mention maybe in 5 minutes at the end, just two unpublished ongoing studies that we have looking at how variation between human individuals, between human populations in the microbiome can regulate host genes differently. And also mention some work we're doing correlating the microbiome and host gene expression and health and disease. Okay, starting with a little background, I think this is the first study that I'm aware of that actually looked at association between host genetics and the microbiome. Didn't actually look at genetics, but just relatedness between individuals. It's shown here on the x-axis. And then on the y-axis, you have the similarity in the microbiome between individuals. And what you see is that there is a slight, maybe significant, maybe not, positive correlation between relatedness and similarity in microbiome, whereby individuals that are more related have more similar microbiome. So this is kind of early evidence, maybe suggestive there's some kind of a now, as the field progressed, there are more and more evidence. It's just an example of another early study where they showed that individuals, or basically twins, so monozygotic twins, with twins that have a completely similar genome, have more similar microbiomes compared to dizygotic twins, although the difference is still not significant. And they also showed in the same paper that individuals from the same family also have more similar microbiome compared to unrelated. So again, more evidence there might be some kind of a genetic effect on the microbiome. Some later studies continue kind of support the hypothesis. This is a work actually from the Bordenstein lab showing that there is an association between the microbiome and ethnicity of individuals from the US. This is a self-reported ethnicity. And we actually collaborate with Seth's lab on this project, basically doing kind of a machine learning analysis showing that you can actually predict the ethnicity of an individual just from their microbiome data. So again, they're suggesting there could be a genetic basis to this, although you could also say this could be environmental. People from different ethnicities might be consuming different diets or have different environments that might have an effect. A big reason why we think there is a genetic, or thought there was a genetic factor when we started this, is from studies in mouse, actually. And this is one of these studies. There were many other studies, a lot of them coming from the Blekhman Lab, showing that you can actually use linkage mapping approaches to identify regions in a mouse genome that are associated with the composition of the microbiome. And you can see kind of the outcome of one of these studies. This is the mouse genome, and the chromosomes, each of these areas that are colored is a region in the mouse genome that has been associated with some kind of change in the microbiome. So you can see they found a number of regions in the mouse genome associated with the composition of the microbiome. And this is the first paper from my lab, where we used kind of a similar approach, but more kind of a genome-wide association approach, to identify the sites in the human genome that are associated with the microbiome. So this is using data from the Human Microbiome Project. It had this interesting tidbit, this study, where the Human Microbiome Project actually did not generate any human genetic data. But the fact that they had so many different body site samples from each individual and have such deep coverage, we were actually able to pull all those data together and find the host read, the contamination host reads from the sequencing effort, and basically use those reads that come from the host to look at genetic variation, called genetic variation in the host genome. We used that to do a genome-wide association study, and we found a bunch of genes and regions in the human genome, shown here on the left, that are associated with variation in the microbiome, that's shown here in this phylogenetic tree. I would just want to highlight one result that I think is probably the most interesting, one of the more significant results that we found, and I think the one that has been replicated in other studies the most, which is an association between genetic variation in the LCT locus. So the LCT is a gene in the human genome that encodes the lactase enzyme. I'm sure a lot of you have heard about it if you're interested in human evolution or human genetics. And what we found is that genetic variation in the LCT locus is associated with the abundance of bifidobacterium in the gut. This is really interesting because there's a kind of a obvious connection between the two, because LCT encodes the lactase enzyme, like I said, it's an enzyme that digests lactose, actually breaks down lactose. Bifidobacterium, on the other hand, is a microbe that really likes lactose too. And some studies shown actually likes lactose more than glucose and can grow really well on lactose. So what we think the mechanism is, and this is from a paper by Ruth Lay that I agree with, is basically genetic variation around the LCT locus determines whether individuals are lactase persistent or not. So if someone is lactase persistent, they have an active lactase enzyme. Regardless of their diet, they're always not going to have enough lactose for bifidobacterium. So these individuals are always going to have a low abundance of bifido in their gut. On the other hand, people who are lactase non-persistent, so they have a non-active lactase enzyme, and they still consume dairy products. Then there's enough lactose for bifido to grow on, and then they have a high abundance of bifido in their gut. So it's kind of a combination of host genetics, maybe associated with diet too, like determines the diet, but also changing the activity of a certain enzyme that then can determine whether there's enough food for a certain microbe in the gut. This is a really kind of cool association that I know some labs are really following up on, understanding whether that explains some of the variability we're seeing with kind of lactase persistent and non-persistent around the globe. Another study we've done that I just want to touch on too is looking at genetic variation. In this case, not genetic variation that segregates in the population, but genetic mutations found in cancer. And what we did here is whole exome sequencing in tumors, identified genes and actually pathways that have loss of function mutations in them in the tumor. And we found pretty strong association between which pathways have loss of function mutations in them in tumors, colon cancer tumors, and the taxa, the microbiome, the composition of the microbiome that's found in the microenvironment of the tumor. And we're able to use similar kind of machine learning approach just from the microbiome of the tumor to predict what are the loss of function mutations. So what are the pathways that have loss of function mutations in the tumor? We found some really several cancer-related pathways. This is kind of touching on some of the earlier studies we did on this, showing that there might be some kind of association of host genetics and the microbiome. I also want to highlight a tool that we developed for bioinformatics, so specifically for doing kind of this GWAS of host genetics and the microbiome. It's called Hominid. It's found on our GitHub. It basically takes an input host genetic variation data, microbiome data, and generates a list of host SNPs and host genome. and they're significant for association and also the taxa and the microbiome that are associated. This is using lasso regression, which I might mention later. Okay, so this is kind of a little bit of a background, kind of what we know, what we think about post-genetic control of the microbiome. Now I'll tell you a little bit of a longer story about the heritability of the microbiome. And the first step, I think, is really important to define what heritability is, because there's a lot of confusion. Sometimes people think about heritability as inheritance, so microbes that are passed on between generations. That's not entirely the case. So when we talk about heritability, I mean it strictly as a statistic from population genetics. I'm sure a lot of you have heard. It's basically a statistic that's used to estimate how much of a variation in a trait in the population is due to genetic variation between individuals in the same population. So basically, this is kind of a ratio of how much genetic variation of the variation we see in this trait. So it's usually a ratio between 0 and 1. Traits that have heritability of 1 means they're entirely genetic. So there's almost no non-genetic effect on this on variation of this trait. Traits that have a heritability of 0, it means there's no genetic effect of these traits. These traits are completely controlled by non-genetic, right? And you can look at the heritability of any trait. Anything you can think about, anything you can quantify, you can actually calculate the heritability of that trait. And people have actually been doing that for many, many years. And you can see just this is from a review paper, just kind of a review of heritability in human studies. You can see that things like height, for example, one of the most heritable traits is 80% or 0.8 heritable, meaning it's very genetic, very genetically controlled, very little environment, but there is some kind of an environmental effect. And then you can see like BMI between 0.45, 0.8, and then it goes down to all kinds of like personality traits that have usually kind of lower heritabilities, like neuroticism, 0.13, things like that. And another thing you can notice here is that heritability is not one number per trait. It can change, and it's different based on the population you're looking at, based on how strong the environmental effects are in the specific context. So heritability is not something that's set, and it's the same for that trait. It can change based on the populations you're looking at and based on the context. This is a little analysis I did where I actually went to the UK Biobank data set. So UK Biobank is a big data set with about 1 million, I think, individuals that they had sequenced, and very, very rich phenotypic information. So thousands of different traits that they looked at, and they actually already calculated the heritability of all these traits, thousands of traits. And what I did here is just basically plot the distribution of these heritability values that they have for all their traits. And just kind of just to see, this is kind of the distribution we would expect from when you look at lots of traits. You have some things that are very heritable, like the height and things like that are here. And then you have a lot of those other traits, like personality traits, things like that are somewhere here between 0 and 2. This is kind of what you expect when you look at any kind of trait. So the question you can ask is, why do we want to study the heritability of the microbiome as a trait, right? And I think this is a really important question. So first of all, this calculation allows us to quantify, to basically put a number on how much of the variation in the microbiome is genetic compared to non-genetic, which I think is a really important question for many reasons. I mean, one thing you can think about is, like if you want to, if you know a microbe is heritable, so it's controlled by genetics, it might not be a good target for treatment, right? You don't want to focus on that microbe because it's less environmentally controlled, and vice versa. You could identify certain targets that are very, more amenable to treatment. And another reason, microbiome traits are heritable, that means those traits are visible to natural selection in the whole genome, which means that if you have that specific trait, this microbiome trait, you can have selection of the host genome in response to the differences in that trait. And to me, this is like a baseline understanding, like showing that a microbiome trait is heritable is the first step if you ever want to talk about coevolution. If you don't show that some trait is heritable, any kind of correlation you see between this microbiome trait and some kind of a host trait could just be correlation. So heritability for me is a really important step whenever you want to talk about coevolution between the host genome and the microbiome. Okay, so my lab was obviously not the first one to think about looking at the heritability of the microbiome. This, I think, is one of the first studies that looked at that. So Julia Girdrich was a grad student at the time with Ruth Lay at Cornell. And what she did is, again, use twin data. So we have monozygotic twins, dizygotic twins. If you have higher similarity in a certain microbe in the monozygotic twins compared to dizygotic twins, you have evidence that might be heritable, might be genetically controlled. You can actually quantify the heritability of each taxon in the microbiome. You can see here the tree, microbial tree, and you can see that if you have significant heritability, almost everything is not significant. You can see some parts here that are significant. So you have some evidence that some areas in the microbiome, some taxa in the microbiome are significantly heritable. And this is from a really nice review of microbiome heritability, also by Julia, that shows kind of an overview of what is known about microbiome heritability, kind of summarizing data from across different studies. And you can see there are some studies here. One study actually from Emily's lab, where did really nice work looking into heritability of microbiome traits in the Hunterite populations in the US. And I think there are a couple of take-home messages from this. One, you have a lot of a few microbes that are consistently heritable across studies. One example is this family, Christensen was shown to be heritable across multiple studies. And you can see across studies, it gets like pretty high heritability, like 0.6 in some studies. On the other hand, you have some taxa that are really heritable at 0.5 in one study and then 0 in another study. So there's actually quite a lot of like inconsistency between studies and what studies are finding. The other thing you can notice from here is that the heritability is actually pretty high. You get actually a bunch of microbes that get like to 0.5 and 0.6, which is really high if you consider the distribution of heritability values that you get from human traits. Or in the UK Biobank data. So a little bit high. It seems like a little exaggerated to me when I look at these numbers, especially when you think that the microbiome is changing a lot. The microbiome we know that it has a pretty strong environmental effect. So seeing those kind of numbers seems a little high. And the challenge with that, the reason maybe why some of these inconsistencies is that it's really hard to quantify heritability of the microbiome in humans for a number of reasons. So there are, like I said, strong environmental effects, tons and tons of different dietary effects on the microbiome. This is an example from a study that I really like from Zernakova et al. Basically just quantifying just like very long dietary questionnaires, quantifying the effect of each individual dietary factor on the microbiome. You have things like how often do you eat rice? How often do you eat buttermilk? Stuff like that. And you have and all these things are correlated with specific things in the microbiome. So a lot of dietary effects, and you can have other environmental effects on the microbiome as well, right? It's hard to really control for. And the estimates of these environmental are actually inaccurate, right? Even if you do have this big questionnaire and you give it to study participants, they're unlikely to actually fill it accurately, because I don't know if you've seen those questionnaires. It's like 100 pages with questions about like how much bread did you eat per week in the last year? Who can actually answer that? So they either answer it incorrectly, or they on purpose, or not on purpose. They can't actually answer it. So you have very inaccurate estimates of environmental variables. And another really big issue is longitudinal plasticity. So the microbiome changes really dramatically day by day, right? And it's sometimes really hard to actually get a good understanding of what the microbiome is like if you take one sample. And in humans, it's really, really hard to get multiple samples. Sometimes there are studies that have 2 samples, sometimes 3. But it's really hard to get patients or study participants to come back for more and more samples. So this is something that's really hard to control for in humans. So what do? Instead of humans, we're actually looking at primates, wild primates, in this case, wild baboons. This is a really, really amazing collaboration we have with the Amboseli Baboon Research Project. This is a project that started more than 50 years ago, so in 1971, started by Gene Altman at Princeton. And this study is basically, for the last 50 years, has been doing daily observations of animals, of baboons, in this one specific community, the Amboseli community. And it's been used as a model to study all kinds of things, like behavior, genetics, ecology, aging of these baboons, by basically collecting very detailed information about their lifestyle, their diet, their social interaction, their habitat use, everything. So it's a very, very rich metadata that we have on these baboons that we can kind of control for in our analysis. Most importantly, we actually have very detailed information on kinship. So we have the complete pedigree of all the individuals in the population, which we can use if we want to look at heritability. We can actually know the relatedness between any pair of individuals. We can use that to calculate heritability. Also importantly, they've been collecting fecal samples for the last like 20 plus years from these animals and preserving them. So they've been collecting them mostly for hormonal analysis. We want to look at hormone levels in the feces, but we started thinking about maybe doing microbiome analysis using these stored fecal samples. That was maybe like 13 years ago. It took about 10 years for this paper to actually come out, but this is a really amazing collaboration. And mostly I want to highlight Jenny Tang and Beth Archie, who are the co-directors of the Amboseli project and are really, really amazing collaborators. And there were a lot of people involved in obviously collecting all these samples over 20 years, but also just processing. Imagine you have thousands of fecal samples in the freezer as powder, right? Dried powder. So you want to be able to put them in 96-volt plates. That took like a year of work. And then actually doing the extractions and sequencing and QC and analysis and processing, it's years and years of work. So a lot of people are involved, a lot of them not mentioned here by name. I do want to mention Laura Greeneisen, she was actually a grad student with Beth Archie at Notre Dame and then a postdoc in my lab and now runs her own lab at the University of British Columbia. And you can see her here at the Amboseli right after collecting a fecal sample from the baboon. Please disregard the lack of gloves. And this is just one paper we published before kind of looking at heritability, just kind of as a proof of concept, just showing that we can actually look at data in this population. We can use it for microbiome analysis. This is using a much smaller subset of the samples that we had. But the whole goal for this paper is to look at social interaction between individuals. And what we show here is that we have a complete social interaction network of all the individuals in the population. We know who interacts with who, and we know at what capacity. We can actually use that to quantify the effect of social interaction on similarity in the microbiome, while controlling for genetics, diet, all those things that are really hard to control for a human. And we see there's an effect of social interaction on similarity in the microbiome. Okay, so back to heritability. So we have all these samples. In total, we had more than 16,000 microbiome samples that we included in the study. This is from about 600, a little less than 600 individuals. You can see the samples here. So the x-axis is the age of the individual. The y-axis is just an individual. So each row is basically one individual, and each point is one sample. So you can, and the green is female, purple is male. So you can see there, we actually have pretty deep longitudinal data. So lots of samples for each individual, on average 28, but for some individuals, we have hundreds of samples, basically kind of sampling throughout their entire life, which is really, really nice to kind of be able to control for that longitudinal variation in the microbiome. We included samples that were collected over 14 years, and we used 16S ribosomal RNA sequencing as a first step. That was the best thing to do. We have now completed doing metagenomic shotgun on all these samples just earlier this year, and we just started processing it. Probably going to finish in like 5 years. So next time I come give a talk, there might be a result from that. And like I said, we have detailed information on diet, relatedness, social interaction, and the environment. And if you ever want to see what it looks like a stacked bar plot, 16,000 samples, this is what it looks like. So you've seen stacked bar plot microbiome data before. So each column, each of these very narrow columns is one sample, and the columns represent different taxa. There's really not a lot you can learn from this, except for a couple of things. One is that there's a lot of variability in the data, right? Which is important if you want to look at heritability. If there's no variation, there's really not a lot you can do. So there's a lot of variability, which is good. And then you can see it's a huge data set. And also something I like to say that you can learn that sometimes you can use microbiome data to generate art. And this is what the same data looks like when it's ordered by the time when a sample was collected. And the take-home message from here, first of all, it looks like a regular microbiome data from baboon. Probably we've looked at this population and other primate populations before. Looks okay. Another thing you can see is that this kind of cyclical pattern is like goes up and down like once a year or so on average. And this is because of the seasonal differences in diet. So you have a wet season and a dry season. The wet season is much more dietary variation, a lot more food options, and the dry season is much less. And you see those cyclical annual changes almost in the microbiome. Again, this is something we can control for, which is good. Okay, so what do we want to do? We want to look at the heritability of the microbiome. What Laura did here is basically define these microbiome traits, microbiome phenotypes from this data. She went really, she did whatever she could, any kind of trait she could define from this data she did. So looking at 7, what we call community phenotypes, those are principal components, richness measures of the microbiome, then hundreds of taxa where we look at their abundance levels at the phenotype, and then more hundreds of types where we look at the presence absence of the phenotype, like a binary phenotype for those taxa. And she basically ran a model that calculated the heritability of each of those 1,000+ phenotypes separately. And the model is basically controls for all these confounders, all these effects that we can think about that are not genetic. So basically the age, the sex of individual, the full social interaction network, maternal effects, if you just have a certain mother, that could have an effect that you control for that in your data. Technical effects, whenever you have a big genomic data set, there's always going to be technical effect there. We control for all of those. And environmental effect, importantly, what's hard to control for humans, things like diet, rainfall, the season, and so on. And when I say we have the detailed dietary data, this, what you see here, is the dietary information that we have. So this is also arranged in the stacked bar plot, but this, instead of microbes, it shows the abundance of different food components in the diet. So you can see here, again, the same cyclical pattern that you saw in the microbiome data, but now you can see why. And the reason is that in the dry seasons, those kind of big blue areas is where the food is mostly composed of corms. And that's the dry season. Again, we have this information, we can actually control for that for the specific dietary composition at the time sample was collected. Okay, and what we're finding, what Laura found here is, big surprise, basically all the microbiome, or essentially almost all, about 95% of the microbiome traits you looked at are significantly heritable. So whether you look at relative abundance of microbes, whether you do certain types of normalizations of the microbe, whether you look at presence absence traits, all of them are significantly heritable, aside from a very small proportion that have non-significant heritability. It's a little bit of a surprise for us. But another thing you can take away from this is that the heritability is actually pretty low, right? So on average is like less than 0.1, and the distribution kind of goes from 0.1. The highest is like 0.2 or so, right? So you basically, everything is significantly heritable because we have big enough data set, we can actually calculate small effects. And the effects are pretty small. So not a very strong genetic effect. And if you think about this distribution that I shared before, of heritability of like many different human traits, I think this looks pretty similar, kind of, where almost all of the traits, any kind of thing you think about, mostly are in this range of between 0 and 0.2. So almost any trait you can look at human has some kind of a genetic effect, but the genetic effect is relatively small. And we don't really see in the microbiome a lot of things that are in that range, where it's like very strong genetic effect, at least in our study, the baboons. You can look at also specific taxa and the heritability levels of these taxa. These are the most heritable taxa. The most heritable trait that we looked at is actually Brachyurtus PC1 of the microbiome. And then you have some taxa, the most heritable taxa is Prevotella, specific ACV within Prevotella. And then it goes down. The ones in red are some that were highlighted in some human studies, like Christensen and Lachier. I mentioned it before. It is heritable in humans. It's one of the most heritable in humans. And we see it as one of the top most heritable taxa in the baboons as well, which prompted us to kind of think about, do we see similar patterns in overall correlation of the of traits? And we basically compared the heritability we're finding across all the taxa in our study, the baboon study, and what other people have been finding in human studies. And we see there's a correlation. It's not very strong, so I don't want to overinterpret it, but there is a correlation, maybe suggesting there might be some kind of a shared genetic basis to the control. Another thing we can do is actually split the data into two. I mentioned the 2 seasons, the wet and dry season. We're going to split the data into 2 and calculate the heritability in the dry season and in the wet season. And what we're finding is that there is a strong correlation between the 2. So any taxon is correlated, the heritability in the wet season is correlated with the heritability in the dry season, but it's consistently higher in the dry season, which means that basically because there's less environmental variation, much less environmental variation, there's more space for genetics to have a bigger effect. which is also highlights how heritability can be context specific. So different seasons have different heritabilities, and we can clearly see why. Another thing you can look at with this data set is the longitudinal information. So this just looks at longitudinal changes across individuals. So each row here is an individual in two things. One is the breaker, the species one of the microbiome, and one is just the Christensen ratio, which is the most common heritable human microbiome taxon. And what you can see is, although these two things are pretty heritable in our study, they're still changing quite a lot. So you can see a lot of changes over time, which also highlights one important thing, that if something is heritable, it doesn't mean that it's constant. And you still have a lot of longitudinal changes, and you still have to control for these longitudinal changes when you look at the microbiome. The last thing we wanted to look at is maybe we can kind of explain why this kind of discrepancy in some of the human studies. Maybe it's because of sample size. We have a very large sample size. Most human studies had much smaller sample size, hundreds, maybe 1,000. So what we want to do is like basically subsample from our data and see what kind of heritability values we're getting. And if we take smaller samples from our study, we're getting much higher heritability, not higher, but a huge kind of range of heritability values, which might mean when you have a small sample size, you get high heritability values just because of noise. And once you get in more and more and more samples, then you get a much tighter distribution and more accurate quantification of heritability, which again, we're finding is like above around 0.1. Another thing we can see is like most human studies found that maybe a handful of taxa are actually significantly heritable, maybe 5 or 10. We found that almost all of them are heritable. And we see this is also because of sample length. When the bigger sample size you have, the more likely you are to actually call the correct amount of taxa as heritable. Again, these are small effects, so you need large sample size to actually quantify. And the last thing is look at the effect of longitudinal data. What we see here, the more samples we have per individual, again, the more likely we are to call the right percent of heritable taxa. And I think the take-home message for me, to you, for anyone working on microbiome field, not just in the heritability context, but any context looking at microbiome data, is the longitudinal data. I think we're going to find it really, really important. And actually, having many samples is the only way you can actually control for those really strong dynamics you see day to day in the microbiome. And without that, you might get a lot of spurious results, which I think we're seeing a little bit in the future. And also just to mention that we kind of, when we worked on this, we realized there's actually no good way to kind of visualize longitudinal data. So we developed this really nice R package just for visualization of longitudinal data. So if you have longitudinal data in the microbiome, it's on our GitHub, you can download and kind of plot that using this horizon plot approach that shows kind of changes over time of certain taxa. Okay. We don't have a lot of time, so I'll go kind of pretty quickly through my next story, which is looking at global variation in the microbiome. I'm going to highlight two earlier studies from my lab, Morton et al. And Gomez et al. From two former postdocs in my lab. I just found out that Andres Gomez, the postdoc that wrote this paper, got tenure just a few weeks ago. Great, amazing, very happy. And what we've seen here is basically when we look at different human populations, we see pretty big differences in the microbiome, lots of changes across populations, but also within the population based on the subsistence patterns and the lifestyles of individuals. So hunter-gatherers have very different microbiome compared to farmers and kind of more industrialized individuals, especially if you compare to the US. We see pretty big differences in microbiome. Not from my lab, other labs have looked at this. It's just a couple of examples. The interesting thing, this paper came out a couple of years ago from the Sonnenberg lab that shows that those differences in the microbiome between industrialized, non-industrialized populations actually has an effect on disease and host phenotype. These are actually important differences to look at and study. And the problem we have in the microbiome field is that our sampling or the information that we have is really not equally represented of different populations, not equally representative of the different populations. So it just shows, we basically went through SRA, so all the microbiome data is available. We didn't actually look at the data, but just where the samples came from. And what we see is that there's this kind of huge enrichment in samples from Europe and North America, which is in this pink block here. So although this is only like 15% of the world population, we have like 70 or 80% of the data coming from this population. And if this figure looks familiar, we're really inspired by this figure, where they've done a similar thing for human genetics, look at the human genomes and their ancestry, and found a very similar pattern where like 80% of the data is coming from people with European ancestry, and very, very few human genomes are coming from other populations. However, in the human genetics field, there are actually lots of efforts to do this kind of global sampling and to sample more population. Things are like NIH-funded big projects like the Human Genome Diversity Panel, HalfMap, 1,000 Genomes, and more recently H3 Africa are really aimed to, by design, collect as many samples as possible from multiple populations. There's really not a lot of these kind of efforts in the microbiome field. One way you can solve this is to actually look at just publicly available data and integrate all of it. So there are petabytes of data at the SRA. You can see how much data is available. You're probably familiar with SRA. Whenever you publish a paper, you have this annoying step of like spending a day and submitting your data to the SRA, and you don't like it, and then the NIH has to maintain this database and pay for it, and they don't like it. So they had this call for like, can anyone actually do something with all this data that we have that we're paying to keep, that no one likes submitting? So we decided, well, maybe we can use that to actually look at global variation in the microbiome and combine it. And we know people have done that in the other genomics field. Like, for example, this is an example from the RNA-seq field, where our collaborator, Casey Green, basically downloaded all the RNA-seq publicly available data and integrated that into one big matrix where you have like all the genes and the expression level across all samples, and got some really interesting biological insights from that. And microbiome data is actually really amenable to this kind of analysis, because as you focus on 16S, you all know what 16S is. I don't have to explain it to you, but it's a much smaller data sets compared to what you have in RNA-seq, where you have tens of millions of reads per sample. With 16S, you have like, I don't know, tens of thousands of reads, very few that have more. So it's actually pretty doable, what we thought when we started this project. It's doable to do the same thing with the microbiome, and this is how we built we call the Human Microbiome Compendium. Again, this is the goal, to integrate all the publicly available human microbiome data. Our starting point is just 16S data of gut microbiome, so only gut 16S data. The next steps, which we already started working on, is looking at other body sets and maybe also integrate metagenomic data. This is the pipeline we use. This is all led by Rich Abdul and Samantha Graham in my lab. This is one of those slides where it's like 2 years of work, and I'm going to say, well, this is what we did. And we got eventually a compendium with 168,000 samples that we integrate. So this is one big table with like 168,000 rows of all the samples, and the columns that represent the taxa. And you can download that. It's all available in our Human Microbiome Compendium website. I'm going to specifically talk in the next few minutes just about our analysis of the data set, where we kept only 150,000 samples for this specific analysis. Some very kind of boring things you can do with this first is just look at how many reads you have per sample. Like I said, about 40,000 reads, but you do have 16S data sets with like tens of millions of reads as well. You can look at how many unique taxa you're detecting. The more samples you have, which is identified if you focus on the genus level or the order level, for example, which is in magenta here. When you get to like 10,000, 50,000 samples, you're not really detecting anything new, no matter how many samples you're adding. Genus level, you're still detecting some new things, but the rate is really getting low when you have a lot of samples. You can also look at the taxa you're finding there. Nothing really interesting here. Just one thing I think is kind of interesting is that you have some taxa like lactobacillus, vectoridalis, where almost any sample, like maybe 80% of the samples that we actually have this microbe. So some microbes are really, really common across human population. This is a stacked bar plot. So we had 16,000 samples. This is 160,000 samples. Again, I think the interesting thing here is that if you look at the blue, I think the blue is Firmicutes. You have this huge variation between individuals, where like some individuals have 0% of this taxon, some individuals have 100% of this taxon, and you have some other taxa that have very similar distribution. So lots of variation between individuals that I think is really interesting. We can recapitulate the analysis we've done before to look at which regions in the world. So we separate the world into 7 regions based on the UN designations. And what we found, again, we basically combined North America and Europe. based on this UN designation. You see again about 80% of the data is coming from Europe and North America, very little from other world regions. And you can do this kind of fun thing, redraw the world map and basically size the different regions based on how many samples come from there. Again, it's kind of funny, but also kind of sad in a way, because it tells you that like almost everything we have is oversampling North America and Europe, and some content, full content is like disappear completely from this map, like South America, Sub-Saharan Africa. You can look at variation of data that way. I want to just skip that, just tell you basically, take home message, there's pretty big differences in the microbiome between world regions, and you can kind of quantify them, those differences in different ways. I'm going to skip that. The take-home method, I think, from this study is that we can basically do the same analysis of like detection rate of taxa, but now separated by world region. So if you look at Europe and Northern America, you can see that we're still detecting new taxa, but at a very low rate. So we can sequence more samples. There's not a lot new that we're finding. However, if you look at regions like Sub-Saharan Africa, which is in orange here, North Africa and West Asia, which is this light orange, really hard to see it because there's so few samples that gets to here. But we're not even close to like detecting all the taxa that we can detect in this region. There's a lot more to find if we sample from some of these sub-sample regions. Another way to look at this is how many taxa we're detecting per million reads at this point. So again, Europe and Northern America, we need several millions of reads to actually detect one new taxa. While in other regions, we need a lot less to detect more. We can also look at different specific taxa that are different between UN populations. The take-home message here is that every taxon we looked at is different between at least the two pairs of human of region, global region. So lots of differences in the microbiome. We quantified using this kind of heat map that shows how each taxon is different between Europe and North America and each one of the other regions. So we can see some of them are like different across the board. Some of them are different in certain populations. Another way to look at it is look at certain taxa and the distribution of the relative abundance in each region. You can focus on Bacteroides here, where we know there's kind of skew distribution in Europe and North America, where there's higher abundances of this taxon across in Europe and North America, while some other populations, like specifically it's the Central Asia here in light blue, where you have much lower abundance of that taxon. Prevotella is kind of the opposite example. We have lower abundances in Europe and North America and higher abundance in some other world regions like Sub-Saharan Africa, for example. We see basically the opposite effect. None of this is really new. So we had ideas that this is all true from other studies. This is the first time I think we're quantifying at that scale, region, and quantify how it looks like when you look at thousands of samples. We also have those same signatures of differences, but at a lower scale. We can actually look at the country each data set is coming, or each sample is coming from, and you can see differences in the microbiome of each country, and the clustering, this kind of country level clustering, is much more significant than you would expect by chance. So you have country level differences. There's a lot more in this paper. This is all available in our website. We have a website called microbiomap.org. You can go to this website. You can definitely download the paper and read it. You can also download an R package. You can actually use to kind of interact with the data, download the data. We have this really nice recipes for the R package. So like, how do I get samples in a specific project? How do I get samples from a specific world region? Can I enrich data with specific country names and so on? So like a way to play with the data and get the specific portion of data you're interested in. And we also have a really nice interactive, I think really cool, where you kind of go to different world regions and like click on different countries and get the distribution of the taxa in that world region and look at the differences that way. Okay, so I think I have like 3 minutes left. I'm just going to mention, I'm not actually going to talk about any results or anything. I've just mentioned that we're really interested in also looking at that direction. So now that we know that there's a huge variation in the microbiome between individuals, between population, we're really interested in understanding how that variation actually affected the host. Is it really important? What does it do to the host? And one way we're thinking this is happening is through changes in host gene regulation. This has been shown in many study model organisms, mouse intestine, zebrafish, stickleback, where you change the microbiome, you get big changes in gene expression in the genes that interface with the microbe. And we think this is what might be happening in humans. So we have a study, an ongoing study, where we're actually trying to do in vitro analysis of those kind of effects of the microbiome host gene regulation. So we use colonic epithelial cells. These are cells that come from one individual and then are basically a cell line. And we extract live microbial communities, so full microbial communities from individuals from different human populations. This is in collaboration with the Global Microbiome Conservancy, Biobank. And we basically look at the effect of these microbial communities on host gene expression using this approach. We can do anything we do. Here we focused on RNA-seq in our first project. We can do methylation, we can do taxi, do whatever kind of genomics you want. And we're basically looking at the effects of how different microbiomes from different populations affect host gene expression differently, and also specific taxa, where the abundance of the taxa is correlated with the response of specific genes. And we have some pretty cool results. I'm not going to go through results. It's still preliminary, so it's still unpublished, but we're getting basically the effect where you see certain host pathways that are downregulated or upregulated in response to the microbiome, and those pathways are different when you look at the industrialized microbiome compared to non-industrialized microbiome. And some of them are disease-related, and we also find specific association with specific taxa in the industrialized, non-industrialized microbiome that drive the expression of certain host pathways. In this context, we also have some really exciting preliminary results that we got yesterday, so I didn't get a chance to include them here, where we actually, instead of colonic epithelial cells, we actually looked at used organoids. There's a colonic organoids grown from specific patients. So because we have multiple organoids, we can actually look at genetic variation within each organoid, and we found some really interesting results of SNPs in each organoid, in each patient, that drive the response to certain microbes in those communities. Very preliminary. I don't want to say more about it because I'm probably going to be wrong and probably going to get different. Really exciting. And I'll just mention that we're also really interested in developing this kind of multi-omic approaches to integrate gut microbiome and host RNA-seq data. This is from a paper by Sabao Priya, a former PhD student in the lab, where we developed these approaches to integrate those data sets in a way that's kind of lets us understand better the mechanisms of interaction between the host and the microbiome. This is also a method that's, I'm not going to go through results, but it's also a method that's available on our lab GitHub. I don't have the address here, but it's the same address as GitHub. It's Blekhman Lab at GitHub. Okay, I'm going to stop here. I think just a quick summary. I think I've hopefully shown you that there is a host genetic effect on the microbiome. It's pretty important to look at, very relevant for disease. The effects are pretty small, but they're pretty consistent across the microbiome. There's big global variation on the microbiome that's kind of underappreciated, and we need to study better. And I described some resources we're developing to kind of try to understand what is the effect of this microbiome variation on hosts. All right, that's it. I want to highlight everyone who's been involved. I think I highlighted specific individuals in their specific studies that I mentioned, all of our collaborators. This is our funding, and that's it. Thank you very much. I'm happy to take questions.
So traditionally, we take our first question from our trainees. So anyone with a question? We're not training.
Nice. Impressive targets. Thank you for that. So for heritability, have you thought about looking at heritability of microbial genes and gene functions?
Yes.
Yeah.
Oh, definitely. I've thought about it, and this is one of the reasons we did the metagenomic shotgun sequencing. So with 16S, we can't really do that.
Yeah.
We thought about doing like prediction, like with 16S, you can do prediction of abundances of certain genes or pathways in the microbiome. We decided not to do it because we're not certain that it's really accurate, those predictions, and we'll see. We're worried the reviewer is going to tell us it's not accurate, and we'll remove it anyway. So we didn't do it. But with the metagenomic data, we're definitely doing it. This is one of the things we want to do. Like, so how does the heritability of pathways or functions in the microbiome? What does that look like? Yeah, I think that's a really interesting.
Thank you. In the study with the primates, I think when you showed, I think it was very interesting when you showed how the heritability changes as you go into the drier months. So I got curious, do you think that epigenetics can play a role in how it modulates the microbiome and how it changes the heritability? in those individuals?
This is a really good question. We have not looked at epigenetics at all. I think it could be really interesting to look at that. Then I think there were studies that shown that changes in the environment can have effect on epigenetics, and then on the response to another change in the environment. So the next time this happens, you're gonna have a different response, maybe less severe response. We don't have epigenetic data from this study. It would be an epigenetic changes in the host. We don't really have host genetic data for the study, so it's all based on relatedness data, everything I've shown. So it'd be a little challenging to look at that specific study, but this is definitely something I'm interested in thinking about in other contexts.
Hi, I was curious about the human host and microbiome across the globe results. Have you looked at what this would look like with admixed individuals, or is it because the heritability of the microbiome is relatively small? Admixture wouldn't really play a role in terms of like how the microbiome communities might differ across the globe.
Yeah, this is a really good question. So those different, the question is whether those differences between populations, is this because of genetics or environment? And we don't really know, right? For this study, we don't have any genetic information. And your idea of looking at admixed individuals to see if that gives you like some kind of an intermediate microbiome, I think that would be nice, and also give us like evidence that some of it could be genetic. In a way, this is what they're doing with the twin study, where they look at the same. But this is really interesting, and definitely something we've been thinking about with some studies where we do have host genetic data, is to look at like the proportion of genetic material basically coming from a certain population, another population, and correlate that, as opposed to just specific SNP, correlate that with the microbiome. Yeah, it's definitely something we've been thinking about. I haven't seen anyone do that. It's a good idea.
I think there was a question online.
Well, it's 12, so we, in respect of everybody's time, if you need to leave, you can get up and leave. There will be lunch in this room, so if you want to continue the discussion, Rand is our hostage for now. To hang out. Let's thank Rand for a fantastic talk.